Okay, guys, so now we're going to do 5.1, and that is the plasma membrane components. Excuse me. Now we're going to be doing plasma membrane components and structure. We alluded to that already. We said the plasma membrane is the fence that surrounds our cell and is a selective barrier. So it allows some stuff to go in and prevents others from entering. What did we call the structure of the plasma membrane? Amphipathic. Can somebody please tell me what does amphipathic mean? It has to be in the head, not in the book. Yeah, in the exam, it's not an open book exam. So amphipathic means that, remember we said it's like a part horse, part human, that's like a mixture, a hybrid. So it has phospholipid heads and nonpolar hydrophobic tails. So the plasma membrane or the cell membrane defines the borders of the cell and keeps it functional. It is semi-permeable, allowing some material to freely cross while restricting others. Who's freely crossing? Who's welcome to go through? The polar or non-polar? We said that in the middle, there is non-polar tails. So please, don't close it all the way. Just like, so if somebody wants to come back. Thank you so much. What is the structure of the plasma membrane? It's a hybrid. The heads are polar and the tails in the middle are. So that means if you're polar, can you cross the middle? You cannot. It will be blocked. But if you are non-polar, can you cross the middle? Yeah. So now is what you got to know. If you are non-polar, like cholesterol-based hormones that are made up of cholesterol, like the testosterone, estrogen, aldosterone, they can cross through easily, no problem. But if you have something like a potassium chloride, Cl minus, Na plus, that's polar or non-polar? If it's plus or minus, is that polar or non-polar? Being plus or minus, does that make you polar or non-polar?
The plus is polar and the minus is non-polar? No.
Both of them. Polar meaning you're one or the other. Polar means you have a polarity. You have a positive pole. You have a negative pole. The battery has a minus and a plus. So if you have potassium plus, chloride minus, that's a polar substance. So can it cross the bilayer leaflet? It cannot. So what does it need to go through? The channel, the tunnel. Excellent. So that's what's going to be able to come in. So certain materials require specialized structures to cross the membrane, and sometimes energy is required. The plasma membrane defines the cell borders and regulates the interactions with the environment by controlling what enters and what exits, what's able to enter and what's able to exit. It is flexible so that cells like the red and the white blood cells, they can change shape to squeeze through the narrow capillaries. They change shape in order to squeeze through the narrow capillaries. The surface markers allow the cells to recognize each other, which is vital for tissue and organ formation and immune system responses. Membrane receptor proteins transmit the signals by receiving external inputs and activating internal response cascades. These membrane receptor proteins transmit signals by receiving external inputs and activating internal response cascades. This is known as the fluid mosaic model. That is very important model to understand. So early models in the 1930s viewed the plasma membranes as railroad for track. So they used to look at it as a the railroad or railroad track or sandwich with protein bread surrounding the lipids. So with advances in microscopy, like the electron microscope, it showed that the membrane's core is a double lipid layer, not a single lipid layer. And in 1972, the fluid mosaic model proposed the membrane as fluid mixture of lipid, protein, and carbohydrate. Who determines the fluidity? The fluidity of a particular membrane. Cholesterol. If you have a lot of cholesterol, you're going to be a little thicker. If you have not a lot of cholesterol, you're going to be fluid. So some cells need to be really fluid, others need to be stiff. So if you're stiff, you're going to have a lot of cholesterol. If you're fluid, you're going to have a little bit of cholesterol. So advances in microscopy in the 1950s showed the membrane's core is double lipid bilayer, not a single layer. In 1972, the fluid mosaic model proposed the membrane as a fluid mixture of lipids, proteins, and carbohydrates. It best explains the plasma membrane structure and function. It best explains the plasma membrane structure and function. So you need to examine this fluid mosaic model. You see, what are these circles representing? What are the circles representing? I'm sorry? The phosphate. The phosphate. That's the phosphate, and these are the lipid tails. These are the lipid tails. You see the lipid tails? These are the phosphate heads. So the phosphate heads, they are facing the aqueous extracellular environment. And these phosphate heads here are facing the inside cytoplasm. Both are polar. The water in the cytoplasm is polar, and the water in the extracellular fluid is polar. Both of them are being touched by the phospholipid heads. But the tails are nonpolar. They make a barrier. Nothing can cross them. So if you are lipid, you can cross right through. No problem. But if you are not lipid, then you need this transmembrane protein. You need this tunnel to allow you to go through. What's the name of these proteins that are on the periphery, on one side of the leaflet, not on both sides? What do we call those? Peripheral proteins. Excellent. These peripheral proteins that are on the outside, peripheral proteins that are on the outside, these peripheral proteins that are on the outside, guys, they are on the outside, so they have always attachment and they have bonding with sugars. That's why we call them glycoprotein. They are sugars and proteins. Sugars and proteins. We call them glycoproteins. The principal plasma membrane components are lipids, phospholipids, cholesterol, proteins, and carbohydrates. The phospholipids have a phosphate-linked head group. Very important to know that. Phospholipid head group, glycerol, and 2 fatty acid chains. Cholesterol has 4 fused rings. So the typical composition is 50% protein, 40% lipids, 10% carbohydrates, though this varies by cell type. That's going to vary by the cell type. Carbohydrates are only on the exterior surface attached to proteins and glycoproteins and lipids, known as glycolipids. The membrane is made up of amphipathic, polar and nonpolar phospholipid molecules. So the hydrophilic, water-loving phosphate-linked head regions interact with the aqueous fluids inside and outside the cell. The fatty acid tails are hydrophobic. These are water-fearing. So you see, you see the glycerol? The glycerol is one, that's the glycerol. 1, 2, 3. 3 carbons. 2 out of the 3 carbons are attached to lipid. Excellent. And 1 is attached to the 2 are attached to lipids and 1 is attached to phosphate. Excellent. So the hydrophilic water-loving phosphate linked head groups, they interact with aqueous fluids inside and outside the cell. The fatty acid tails are hydrophobic. These are water-fearing. This allows the phospholipids to form a bilayer, separating the interior from the exterior aqueous environment. The lipid bilayer has hydrophobic tails facing inward and hydrophilic heads facing outward. So you see here the liposome has phospholipids on the inside, and you have lipid heads on the outside, lipid heads on the inside. But the bilayer leaflet is spread out. It's not like a liposome. The liposome is like a submarine. It's carrying inside it something. But the micelle is one layer that is enclosed. So the micelle has in its core, so an example of a micelle is the LDL, you know, the bad cholesterol, low density lipoprotein. From the outside, it's hydrophilic, but from inside it's hydrophobic. So these are different structures. The bilayer leaflet is amphipathic leaflet, like really spread out. But the liposome is when you have the bilayer leaflet enclosed and encircled and tightened. But the micelle, it is only one layer that is enclosed. So the micelle and the liposome are both enclosed. Only difference is micelles have only one amphipathic leaflet, while the liposome have double amphipathic leaflet. So proteins are the second major component of the plasma membrane, and they serve various important functions. So they can be integral proteins. These are embedded in the plasma, embedded in the membrane, or peripheral, temporarily attached. So you see your proteins? Your proteins here, you have proteins on the outer periphery or proteins that span both layers. So integral proteins span the membrane partially or fully, with hydrophobic regions embedded in the lipid bilayer and hydrophobic regions protruding from the membrane. They are protruding from the membrane. So if a peripheral protein is attached to the exterior or interior surface, the periphery of the membrane, so they can attach to either integral proteins or phospholipids in the membrane. Peripheral and integral proteins can function as enzymes, as attachments for the cytoskeleton, or act as recognition sites. So the peripheral and integral proteins can function as enzymes and as attachments for the cytoskeleton or act as cell recognition sites. So you know the binding of the hormone is like a lock and a key, like a ligand. The hormone is going to bind an extracellular part of the receptor, and that's going to open the channel. That's going to allow for an intracellular signal transduction cascade. So these peripheral proteins are important for recognizing a particular signal. Also, carbohydrates are found on the plasma membrane. So we don't only have phospholipids. What is the major component of our plasma membrane? Our plasma membrane is made up of what more than anybody else, more than anything else? The plasma membrane is made up of what more than anything else? It's made up of phospholipids. If you look at this structure, do you see more phospholipids or do you see more proteins? Do you see more phospholipids or do you see more proteins? So the carbohydrates are always found on the exterior surface of the plasma membrane. They are bound to the proteins known as glycoproteins or the lipids. These are glycolipids in straight or branch chains. So they form the glycocalyx, which attracts water to the cell surface and aids in cell recognition, embryonic development, and in cell-to-cell attachment. So now, how do viruses infect specific cells? So the HIV can penetrate the plasma membranes of helper T cells and the monocytes, while hepatitis attacks the liver cells. That's very interesting. So you have the HIV being able to penetrate the plasma membrane of the helper T cells and the monocytes, and hepatitis attacks the liver. But either way, whether you're attacking a monocyte, whether you are attacking a T cell or attacking a liver, you must be able to bind to the plasma membrane and become endocytosed. So viruses like HIV, they can invade the cells by binding to specific surface receptors, while other viral surface proteins interact with the immune system to induce antibody production against the viral antigen. A major challenge with HIV is that the viral surface recognition sites mutate rapidly, generating new viral variants that existing antibodies cannot effectively recognize and bind to. So the problem is with HIV is that it mutates so frequently that the antibodies cannot keep up with making recognition sites based on the mutation. So your antibodies, they can target known variants of viruses, but HIV mutates so rapidly that your antibodies cannot target it. So HIV compounds this problem by specifically infecting and destroying immune cells involved in the body's response, further weakening the host ability to combat the rapidly evolving virus. We talked about the fluidity of the membrane. So the plasma membrane structure allows fluidity with integral proteins and lipids moving relative to each other. So the membrane will burst if penetrated or if it takes on too much water. The phospholipids have either saturated fatty acid tails that pack tightly together or unsaturated tails with kinks. So the kink, we have seen it here. You see this kink? This is the kink. The kink is caused by unsaturated. This is a saturated fatty acid. It is saturated with hydrogen. It has hydrogen all over. But unsaturated fatty acids do not have hydrogen, and as a result, they have this double bond, and that's an example of a kink. So having a kink is very distinctive because the phospholipids are either saturated fatty acids that pack tightly or unsaturated with kinks that create fluidity. So if you have a kink, you will be able to be more fluid. But if you are unsaturated, you will stack so tightly. So a question would be, a membrane, a plasma membrane that has a lot of saturated fatty acids, is it going to be fluid or is it going to be rigid? A lot of fatty acids. It's going to be very rigid because the kink makes the plasma membrane more fluid. So the more kinks are there, the more fluid the plasma membrane is. How, and also we know that cholesterol maintains fluidity across a range of temperature because cholesterol acts as a buffer system. So in colder temperatures, the membrane becomes solidified. The solidified membranes with more saturated fats. And organisms adapt to the cold by adjusting the unsaturated to saturated fatty acid ratio to maintain membrane fluidity. So that is very interesting that your plasma membrane is so dynamic in the sense that when it is so cold, do we need to have our membrane sticking together or do we need it to be fluid? When it's very cold in hypothermia, do we need the membrane to be more fluid or more rigid? More rigid. So does that mean we're going to have a lot of kink or we're going to have unsaturated fatty acid? You see here, look at this. This is saturated with hydrogens, has no kink. And this is unsaturated, it has a kink. Which one will become more fluid if you have a lot of kink or if you have no kink? If you have a kink, you're fluid. If you have a kink, you're fluid. If you have no kink, you are rigid. So when you're so cold, are you going to be more having a lot of kink in your plasma membrane, or are you going to be saturated with hydrogens and with no kinks? You got to be having no kinks because you want to be saturated and really sticking together. So that's how we adapt to cold. When it's cold, our plasma membranes are going to be unsaturated with a lot of kink. I'm sorry, when we are so cold, our plasma membrane is going to be saturated with a lot of hydrogen so it can stick together, no kink. But when it's hot outside, our plasma membrane is going to be so fluid with no saturated fatty acids. We're going to be having unsaturated fatty acid with a lot of kink so that we can be fluid. So cholesterol is also important in the fluidity. So it acts as a buffer, preventing low temperatures from inhibiting fluidity and high temperatures from increasing it excessively. So if you heat up a plasma membrane, it's going to be more fluid, right? And if that is cold, it's going to be more rigid. So the cholesterol is there like a balance. So when it's too hot, it's not going to allow the plasma membrane to be too fluid. When it's too cold, it's not gonna allow the plasma membrane to be too rigid. It's gonna keep it balanced. This cholesterol is like a buffer. You see where my mouse is? Cholesterol is like a buffer. Cholesterol organizes transmembrane proteins into lipid rafts within the membrane, and lipid rafts compartmentalize processes like signal transduction with signal reception and relay by grouping together related compounds. This is the phospholipid that is the main membrane fabric. Cholesterol is attached between the phospholipid and the two phospholipid layers. Integral proteins, peripheral proteins. So this is a summary. The plasma membrane is a fluid mosaic model comprised of phospholipid bilayer with embedded proteins. So you have to understand that this phospholipid bilayer is a very fluid. And what is the most major prevalent component of the phospholipid bilayer? Who is the most major prevalent component? The most major prevalent component, who's that? of the plasma membrane. What is the major? The phospholipids. Followed by who? By proteins. Followed by, after proteins, followed by carbohydrates, because we find the carbohydrates on the outside for recognition with the immune cells and for the glycocalyx. These are the glycoproteins, sugars with protein. Some membrane proteins span both layers, transmembrane proteins, integral proteins, while some proteins are peripheral. They remain on the outside. So carbohydrates attach to some membrane proteins and lipids, forming complexes that identify the cell. The membrane's fluidity results from the temperature, fatty acid cell configurations, cholesterol abundance, and the mosaic arrangement of proteins and carbohydrates. And that was 5.1. 5.2 is passive and active diffusion. So we know that plasma membranes are, now this is we're going to define the difference between passive transport and diffusion. So the plasma membranes are semi-permeable and others are selectively permeable. What does semi-permeable mean? Semi-permeable membrane means that it allows some substances but not others based on their molecular sizes. But selectively permeable, it blocks some substances while permitting others. So you have to know the difference. What's the difference between semi-permeable? Semi-permeable, that means it's discrimination. It discriminates. It allows some substance to enter and prevents other substance from entering, and allows some substance to exit and prevents some substance from exiting. Selectively permeable means the opposite. Selectively permeable means the opposite. It means that it blocks some substance from entering the cell and blocks other substances from exiting the cell. What's the difference between passive transport and diffusion? So passive transport moves the substance from an area of a high concentration to an area of a low concentration. And a concentration gradient drives passive transport of a substance between the locations. So what does that mean? If we look at passive transport and diffusion, if we look at passive transport and diffusion, if we look at passive transport and diffusion, so passive transport, let's say on one side you have 500 millimoles of glucose. On the other side, you have 300 millimoles of glucose. And you put them in two chambers that are connected like that. Then if you open up this barrier and you make it semi-permeable, it's a semi-permeable barrier. This semi-permeable barrier allows for the passage of what movement? You have 500 on one side, let's call that side A, and 300 on the other side, let's call that side B. What's going to happen now? They're going to go from which side to which side? Is 500 going to go to 3 or 300 going to go to 500? So diffusion is the passage of molecules from area of high concentration to area of low concentration. Area of a high concentration to an area of a low concentration. So 500 is going to move to 300. It's going to move until the whole 500 gets to here. You're going to have 500 plus 300, 800. Is that what's going to happen? Is that what's going to happen? Is the whole 500 going to go to the 300? So what's going to happen? Until they both equal. So 100 is going to leave from this side, is going to be 400, and 100 is going to be added to this side, and that's going to be 400 till they're equal. So we have equilibrium. Equilibrium. Till we have equilibrium. They're both equal. Now when they are equal, is there any more movement? Yes. There is movement that is 1-to-1. So one glucose jump on this fence, another glucose makes up for it there. That's known as steady state. Steady state equilibrium. So they're not locked in. They're moving, but they are in a state of equilibrium. So what drove this equilibrium? We used to be 500 on one side, 300 on the other. What drove this equilibrium is the gradient. You need to have a gradient. A gradient is a disparity. A gradient is a disparity. We have different kinds of gradients. We have gradient that is concentration gradient, chemical. We have a chemical concentration gradient. So you go from 400 millimeter glucose to 100. Then there's thermal gradient when it's 400 degrees Celsius, 500 degrees Celsius, and here it's 300 degrees Celsius. So they're gonna even out at 400 degrees Celsius both. So that's a thermal gradient. Then there's pressure gradient. So when your heart pumps blood, the blood is gonna flow down pressure gradient from where closest to your heart down to the bottom of your legs. So concentration gradient drives passive transport of a substance between the locations. Selective permeability. So the plasma membranes are asymmetrical. The interior does not match the exterior. So some interior proteins anchor the membrane to the cytoskeleton, and the exterior peripheral proteins bind to extracellular matrix elements. So carbohydrates on the exterior surface require substances. You got to know that. Who do my carbohydrates bind? Do they bind the transmembrane proteins? Do the carbohydrates bind transmembrane proteins? No. Do they bind intracellular or inner leaflet peripheral proteins? Do you see carbohydrate here? No. Where do you see carbohydrate? Always on the outer leaflet, on the extracellular side. So they bind proteins on the extracellular side. They bind lipids also. So these are known as glycolipid, a hybrid. Sugar with a lipid, glycolipid. And here is glycoprotein, glycolipid and glycoprotein. We said the plasma membrane is amphipathic, and non-polar and lipid-soluble materials can move through the hydrophobic core. So we said that the non-polar can cross through the hydrophobic core, and the polar substances interact with the hydrophilic membrane exterior, but they cannot pass through the lipid core. They cannot pass through the lipid core. So the ions such as sodium, we talked about that. Sodium has a plus charge, that's a positive. Potassium has a plus charge. Chloride has a negative charge. These are all polar. So they cannot cross the the non-polar bilayer. They cannot cross the lipophilic layer. So you need to know the layer in the middle is butter, is hydrophobic, lipophilic. Diffusion. Diffusion is passive transport, is a passive transport process of a low molecular weight material along its concentration gradient from an area of a higher concentration to an area of a lower concentration. It continues until equilibrium has been reached. You see here? So diffusion is the passive transport process of a low molecular weight material along its concentration gradient. So you see all these red dots here. There is no red dots on this side, and you see all these blue dots here. There is no blue dots on the other side, so both are going to be moving in the opposite direction to fulfill equilibrium. So 3 represents for me equilibrium. So when they reach equilibrium, do they stop? But they're still moving, right? So that's steady state equilibrium. So diffusion allows material movement within the cytosol and through the membrane. You see, through diffusion, we go from where there is a lot through the membrane. So the concentration gradients are a form of potential energy that dissipates as the gradient is eliminated. Dissipates as the gradient is eliminated. So when we'll reach equilibrium on both sides, we're gonna stop the diffusion process. Factors that affect diffusion. So molecules move randomly, but the rate of movement depends on the molecule's mass, the environment, and the temperature. So how does the molecular mass affect the rate of diffusion? How does molecular mass affect the rate of diffusion? So the bigger you are, the more massive you are, the harder is it for you to diffuse across the membrane. Also the temperature. So the smaller you are, the higher the rate of diffusion. The bigger you are, the slower the rate of diffusion. Is that point clear? So if you're gonna cross through the plasma membrane, if you're so big, you will not be able to cross. So the bigger you are, the harder is it to cross. So the diffusion rate is gonna be slow if you're so big. But if you're so small, you're gonna have a high diffusion rate. What's the other concept? That is the temperature. If it's so hot, if the temperature is so high, you're gonna diffuse more or less. If you heat up the substance, is it gonna move more or less? It's gonna move more. So temperature is going to increase the rate of diffusion. The dynamic equilibrium, there is no net movement. This is what I mean by steady state. This is a steady state dynamic equilibrium where there is no net movement of molecules despite the constant motion. So we diffuse by filtration. So filtration is when hydrostatic pressure is powering the movement of solutes across the bilayer leaflet. So filtration is a diffusion through a membrane, sometimes using pressure to increase the diffusion rate. In the kidneys, the blood pressure forces water and solutes, dissolved substances, out of the blood. The high blood pressure can force too many proteins into the urine. The high blood pressure can force too many proteins into the urine. The facilitated transport is the process by which material moves down a concentration gradient using integral membrane proteins and no energy. So that is facilitated. So for example, we use that for glucose. Glucose is so big, it cannot pass through the bilayer leaflet. Glucose is so massive, it cannot pass through the bilayer leaflet. So we need a transmembrane integral protein. We talked about that earlier. What did we call this transmembrane protein that's going to allow the passage of glucose down the plasma membrane? GLUT. Remember we said GLUT? GLUT are glucose transporters. So glucose transporters allow for the entry of glucose down concentration gradient through these channels, through these protein tunnels, through these integral transmembrane proteins. So they must first bind to exterior receptors, enabling the removal from the extracellular space and their admission into the inside of the cell. There you go. This is glucose outside. You see, glucose is outside. Facilitated diffusion means you still diffuse from where there is a lot of glucose outside to where there's a little bit of glucose. And you see that there's influx of glucose down this glucose transporters. Same thing with water. Water is polar. So can water go through the bilayer leaflet? So it needs a tunnel. Water needs a tunnel to flow through. That is known as the aquaporin. Aquaporins are channel proteins that allow rapid water transport. Okay, gated ion channels opened by the attachment of an ion channel or ligand molecule or voltage change The kidneys have both gated and open channel for ions. Neurons have gated sodium, potassium, and calcium channels that maintain the concentrations needed for the electrical signal. The carrier proteins bind and facilitate the movement of specific molecules across the plasma membrane by changing the shape, They specify for a single substrate or a single substance. So these are carrier. Like for example, the carriers for the electrolytes. Some protein channels, they carry sodium, they carry chloride down its concentration gradient. But you must utilize a carrier. While osmosis is the movement of water across a semi-permeable membrane that is driven by the concentration gradient of water. So the osmosis is different than diffusion. In osmosis, let's say again, we have 500 millimoles of glucose. And on the other side, we have 300 millimoles of glucose. And we add to them 1 liter of water. 1 liter of water. Is that clear? 1 liter of water. Can you picture that? Can you picture that we have side A and side B? Side A has 1 liter of water and 500 millimoles of glucose. Side B has 1 liter of water and it has 300 milliliters of glucose. Can you picture that?
Yeah.
Now the issue is, I have a semi-permeable membrane now. What does this semi-permeable membrane allow the passage of? What does it allow the passage of? Before, it was just 500 millimole of glucose, 300 millimoles of glucose, right?
Yeah.
So glucose went from high to low because that membrane was allowing the passage of who? of glucose. But this is not allowing the passage of glucose. This is allowing the passage of water, because we added to water. So glucose cannot pass. Glucose too big. We make the pores too small, too tiny. So now what happens to the water movement? What happens to the water movement? The water is going to flow from where the 500 is into where the 300 is. So now water is going to go down here and it's going to go really up here. What is going to shift from where there is a lot of solute to where there is a little. So in osmosis is movement of water, solute, movement of water from area of high, from an area of high water concentration and low solute concentration to an area of low from an area of a low solute. It went from an area of a low solute concentration to an area of a high solute concentration. You see? So we went from an area of a high, the water went from, oops, that's wrong. The water is going to go from an area of a low solute to an area of a high solute. So now the water is going to go from the 300, it's going to drop so low, and it's going to go up so high in the 500. Does that make sense? Osmosis is the movement of water from high water and low solute, 300, to an area of high solute concentration. Why? Because water wants to dissolve it. Remember we said the water is going to dissolve NaCl, and water molecules are electronegative oxygen, electropositive hydrogen, and here water is, so here you have chloride binding with this hydrogen, while here you have sodium binding with the oxygen. So water dissolved, right? For solvation. Water is important for solvation, dissolving. So the water moves from an area of low solute to area of high solute. Let's say I make another side here that now had a 1,000 millimoles of glucose, and I make it permeable to water. Now all the water gonna move here until we reach equilibrium, osmotic balance, until we have osmotic balance. Are we clear? The aquaporins facilitate water transport. They play a major role in osmosis in the red blood cells and kidney tubules. So the mechanism of osmosis is the solute cannot cross the semi-permeable membrane, but the water can. So the solute cannot cross the semi-permeable membrane, but the water can, creating a concentration gradient that drives osmosis. Is this point clear? Is this point clear? The solute cannot, we did that now in the board. We said that the solute cannot pass through the semi-permeable membrane, but water can, creating a concentration gradient that drives osmosis. Osmosis will continue until the water concentrates are equal on both sides of the membrane, or until the opposing forces reach equilibrium. You see what happened? So water moved from an area. You see here you have a little bit of solute, and here you have a lot of solute. So water moved from where there is a little bit of solute to where there is a lot of solute. You see? It dropped and went up here. Tonicity is a measure of the effective osmotic pressure gradient, or the difference in water potential of the two solutions. So osmolarity defines the solution's total solute concentration. So low osmolarity has high water concentration, low solute. High osmolarity means it has high solute concentration and low water concentration. Now let's go over hypotonic, hypertonic. And this is very important for the blood cell. This is how the athletes die. That's how athletes die when they run the marathon. When they run the marathon and drink too much water. So now let's look at a red blood cell. Let's say that we have a red blood cell. That is a red blood cell. I put it in solution A. That is a red blood cell. I put it in solution B. That is a red blood cell. I put it in solution C. That's a red blood cell. That's a red blood cell. That's a red blood cell. I know the osmolarity inside my red blood cell is 300 milliosmoles. Inside this red blood cell is 300 milliosmoles. And in C, it's still 300 milliosmoles. But for solution A, I dip it in a solution here, in a water solution that has a concentration of 300 milliosmoles. So the inside osmolarity is equal to the outside osmolarity. Are you guys paying attention? Ilham. 300 milliosmoles inside the cell and 300 milliosmoles in the solution. What do you think is going to happen to the water movement? Is water going to go from out to in or from in to out?
Is it a steady state?
It's going to be in a steady state. So water going to go a little bit in and then it's going to go equally out. That's isotonic. That's isotonic solution. Okay, so the size of the red blood cell is going to be the same. You get it? Now for B, let's say I put it in a 3,000 milliosmoles. So the inside is 300. The outside is 3,000. How is that going to affect?
It's going to go into the red blood cells.
What did we say osmosis does? Osmosis, water moves from where to where? The water is gonna, no. The water is gonna dissolve. We said when we put the glucose like that, we said you have 300 on one side, 500 on the other. We said water is gonna drop 300, it's gonna go up to the 500. We did it here. If you look at this figure, look at this figure right here. Look at the direction of the water. Did the water move from where there is a lot of solute to where there is a little, or from where there is a little bit to where there is a lot? So you see the water shifted down. Please look there. The water dropped down where there is a little and went to a lot. And we did the NaCl to show that this is gonna mean it's gonna go dissolve. That's osmosis is to dissolve. So here you have 3,000 on the outside. 300 on the inside. What's the direction of water?
It's.
Why would you say equal?
No, it's.
Here, hold on, please hold on. Hold on. We said 300 inside, 300 outside, they're equal. 300 inside, 3,000 outside, why would it be equal?
No, it's gonna go towards the 3,000. So.
Why would it go inside?
It's supposed to come out.
No? You know? Look there one more time. Look here one more time. Look at this figure. Look at this figure. You have how many glucose here? 1, 2, 3, 4, 5, 6, 7, 8. Over here you have like 25. The water moved from where there is a little to where there is a lot. So you see water went from the little side to a lot. We're not talking about glucose. Glucose is going to go from 500 to 300 till they equal. But this is not glucose. This is water. This is osmosis. This is not diffusion. This is osmosis. So now the water is going to go from, so now you have 3,000 outside, 300 inside. What's the direction of water movement? Going out. So the water is going to go out. So what's going to happen to the red blood cell? It's going to shrink. It's going to shrink. Now here you have 300 on the inside and 30 milliosmoles on the outside. What's the direction of the, yeah, it's going to go from where there is a little to where there is a lot until it's going to burst. It's going to burst. It's going to lyse. It's going to lyse or burst. So let's name the solution. That's isotonic solution. That is a hypertonic solution, and that is a hypotonic solution. Okay, do you see the isotonic solution? In isotonic solution, there is equal flow. In hypertonic, water is moving out. Water is moving out. In hypotonic, water is moving in. You see what happened to the red blood cell? In a hypertonic solution, the water is going to shrink. In a hypotonic solution, in isotonic is equal, steady state. But in a hypotonic solution, it's going to burst, swell. So you see what happens with we went over that, the hypertonic, isotonic, and hypotonic. Okay. So this tonicity is found also in the primesia and the amoeba that lack the cell walls and they have contractile vacuole. They collect excess water from the cell's interior. They pump out collected water from the cell. They prevent cell lysis from taking in too much water. So you see, freshwater fish live in an environment that is hypotonic to their cells. And salt water fish live in an environment that is hypertonic to their cells. So you see that hypotonic, the water always goes inside. And hypotonic, the water always goes to the outside. So which one is better, fish? The one that live in the fresh water or the salty water? Why? Freshwater fish. Does water go inside it or leave it? It diffuses inward. It diffuses in the cell. So because the water is fresh, the fish is too salty, so water go inside the fish. So the fish rich in water, so that's why it's good. But that one, water leaving it, so it's always full of toxin, always full of that kind of stuff. The kidneys regulate the amount of water in the body, and osmoreceptors in the brain, they monitor the blood solute concentration. If solutes increase beyond normal range, a hormone is released to slow the rate of blood loss, of urine and water loss in the urine. High albumin protein concentration in the blood helps to control the osmotic pressure and prevent fluid leakage into the tissue. And this is our summary. Diffusion and osmosis move small molecules across the membranes from areas of high to area of low concentration. Each substance diffuses independently based on its own concentration gradient. Diffusion rates depend on the temperature, the particle size, and the gradient. If you're 500 on this side, 3 on that side, water will move from where there is 300 to 500, and the molecules will move from 500 to 300. And you see that in the cell, the concept is the same, but you need tunnel proteins to allow you to go through. Okay, so we will do 5.3 and we will stop here. And then next week we will do 5.4 and 5.5. The exam is going to be up to 5.3, what we're going to do now. So we're going to do 5.3, and the homework that I'm going to publish for you is going to go up to 5.3, and the midterm is going to be up to 5.3. Then after that, we're going to continue 5.4, 5.5, again with next week's chapter, and that's going to be part of the next exam. So we're going to defer 5.4, 5.5 to the next exam. So let's do 5.3 now. So 5.3 is now showing us active and sodium potassium pump. So you know we have two kinds of transport. We have two kinds of transport. We have passive transport that does not use chemical energy. It does use kinetic energy. So the transport of substance, the transport of substance, the transport of the substance is either passive diffusion, or active diffusion. Passive diffusion, active diffusion. You must also know that we talked about passive diffusion being divided into two parts. We have passive diffusion that is facilitated like glucose, facilitated passive diffusion, and that's like glucose. If you have a plasma membrane, you need a glucose channel. You need a glucose channel. You need a glucose channel. You have a glucose channel that's going to allow the movement from too much glucose. There's too much glucose on the outside, and there's too little glucose. So glucose is going to go through this channel known as GLUT channel, glucose transporter channel. You're going to move in. So that is facilitated. Why is it facilitated? Because it did not go through naturally. You needed this membrane channel here, this facilitator. You needed this facilitator, this glute. You needed this glute facilitator to allow the passage of glucose downward. Are we clear? Are we clear? Then we have non-facilitated diffusion. And that is when you just diffuse down concentration gradient without a facilitator. That is non-facilitated diffusion. And that you diffuse without a facilitator. So you diffuse without a facilitator, and you diffuse down concentration gradient. So this is passive diffusion is always down concentration gradient. Down concentration gradient. Are we clear?
Yeah.
You can either do it with a facilitator channel or without a facilitator. Naturally, through the membrane, without a facilitator. But nevertheless, it's down concentration gradient. An example of that would be diffusion or water, osmosis. But in active transport is the opposite. Passive is down concentration gradient, while active is against concentration gradient. This is downhill. Passive diffusion is downhill, while this active diffusion is uphill. This active diffusion is uphill. Active diffusion is uphill. So this is an example of that would be the sodium potassium ATPase. The sodium potassium ATPase there is a lot of sodium on the outside, and there's a little bit of potassium on the inside. So there's a lot of potassium on the inside. So what does the sodium potassium ATPase do? The sodium potassium ATPase supposedly, because there is a lot of sodium on the outside, there is a little bit of sodium on the inside, there is a lot of potassium on the outside, there is a little bit of potassium on the outside. What should be the direction of flow of potassium? Potassium is going to go from where there is a lot on the inside. This is the inside of the cell. This is the inside of the cell. And this is the outside of the cell. So from the inside to the outside of the cell, you're going to go, that is an example of diffusion. Are you clear? In diffusion, we go from an area of high concentration to an area of low. We have a lot of potassium inside the cell, a little bit of potassium outside the cell. So we're going to go from where there is a lot of potassium inside to where there is a little bit of potassium outside. Clear? Then we have a lot of sodium outside, a little bit of sodium inside. So we go from where there's a lot of sodium outside to where there's a little bit of sodium inside. The sodium potassium ATPase doesn't do that. It does the opposite. For every sodium that flows down concentration gradient, she will pick it up, throw it back out. And for every potassium that leaks out, it will take it, brings it in. So naturally, there's a lot of students on the outside. They want to come in, they come in through the door. Imagine there's a bodyguard here. There's a lot of students outside. Those who walk in will grab them, throw them out. So that's what the sodium potassium does. It does uphill, not downhill. It's a violation. And because it works uphill, it has to be really strong, and it uses ATP. It uses ATP energy for that. It uses ATP energy for that. So active transport requires energy and often ATP to move the substances against their concentration gradient. So passive transport is like the movement of water down the waterfall, where the active transport is like movement of water up the waterfall. There we go. If you see here, we talked about that. Look at where is there a lot of sodium. Please look where my mouse is. Where is there a lot of sodium? Is there a lot of sodium outside or a lot of sodium inside? Outside. So the sodium moves in. You see the blue gradient is to go in. Chloride. Is there a lot of chloride outside or inside? So a lot of chloride is going to flow from outside to in. And potassium, is there a lot of potassium outside or inside? So it's going to go from in to out. What the sodium potassium does, it's going to violate that. Once they get out, it's going to bring them in. And when they go in, it's going to bring them out. So if you see here, this is the pump. Sometimes we move against the gradient. We use a pump. This is known as primary active transport when we directly use ATP. When we use ATP to power the movement uphill. So you see there is 4 hydrogen plus, and there is 1, 2, 3, 4, 5, 6, 7, 8 outside. We use ATP. Do you see the ATP here used to pump out the 4 against the 6? So if you use ATP, that's known as primary active transport. But if you don't use ATP and you hitch a hike, somebody else is driving a car, you take a ride with them, that's known as secondary active transport. It uses co-transporters, pumps that move 2 substances at once to couple the movement of a substance down its concentration gradient. The carrier proteins for active transport. You see, this is a uniporter. It carries 1 type of molecule. Symporter, 2 down the concentration gradient. And antiporter, 1 on. This is an antiporter. So 1 goes in and 1 goes out. So an example for that is the sodium-potassium ATPase. It'll send sodium out and it'll bring potassium in 2 opposite directions, making it an antiporter. So you see the primary active transport? In primary active transport, I always utilize ATP. That's the distinction of primary active transport. Using ATP to throw out 3 sodium and bring in how many potassium? 2 potassium at the expense of ATP. You see, that's my sodium-potassium ATPase. It takes, you gotta know this, it takes 3 sodium from inside to the outside, and it brings in 2 potassium from the outside into the inside. It creates a chemical gradient with more sodium outside the cell and potassium inside. Establishes a membrane potential by making the cell interior negatively charged and the exterior positively charged.
Is it always like that?
Yeah.
The inside negative and the outside positive?
Yeah, it keeps it like that. Okay, so we talked about the sodium, potassium. So this is really important for you to see that we bring 3 potassium outside the cell and we take 2 potassium from the outside, bring them into the cell. And this sodium that was sent out, so this is primary. What is primary transport? Primary transport is when you use ATP. And sodium goes out, then this sodium can be used, remember, to hitch a hike, take a ride with somebody else, to bring in glucose. So glucose is hitching a hike with sodium who's going in. Sodium is going in, potassium is coming, glucose is coming with it. That's secondary. You see primary up here, where my mouse says primary, use ATP. But secondary does not use ATP. Secondary uses a gradient of sodium to bring in glucose. So this is the GLUT transporter using the sodium. And we said secondary active transport is when you use an electrochemical gradient of hydrogen protons across the inner membrane of the mitochondria. So here you're hitching a hike, you're hitching a ride with somebody else. So here there is an electromotive force, and with this electromotive force, you are able to transport in a certain molecule. Or with sodium flowing in, you move in with it another molecule. So the combined gradient that affects an ion includes its concentration gradient, an electrical gradient, positive ions, phase concentration gradient favoring diffusion, and in aqueous solutions, both electrochemical and concentration gradient need to be considered. We move the substances against electrochemical gradient that requires ATP, primary or secondary if we have another form of an ion or a force that we take. And cells require some substances at higher internal concentration than external. So that's where our